Good evening, everyone. Good evening. Well, that was good. Very nice. Thank you for being here. This is a great night. I am so happy to see all of you here, to see a number of you out in the reception beforehand, and to welcome you all here. My name is Jim Saylor. I'm the Vice President for Corporate Affairs at the Population Council, hosting this event. And on behalf of all of my colleagues at the Pop Council, as well as particularly our colleagues at our Center for Biomedical Research, which is located on this fabulous campus here at Rockefeller University, I am very honored and delighted to welcome all of you to the second annual Sheldon Segal Lecture. Shelley Segal was not just a tremendous inspiration to the scientific community. He was a close friend to many of you in the audience this evening. And I know that Shelley, and it's Shelley's legacy that we're honoring here tonight, would be delighted to see so many friends and colleagues come to this lecture, which is the first one. We alternate with the Woods Hole, and this is the first one here in New York City. So this is a very special one and an exciting one for us tonight. Shelley would be exceptionally pleased that Dr. Joan Ruderman, who's the Executive Director of the Woods Hole MBL, is here tonight. The Woods Hole MBL is our co-sponsor for this series, and as I said, we alternate holding it in Woods Hole and in New York. Shelley served as the chairman. In addition to having worked at Woods Hole in the summer for many years, he was the chairman of the board of the MBL, and he was an advisor and a counselor to a series of executive directors of the MBL. So we're happy, and he would be very delighted that Joan Ruderman is here. Shelley would be thrilled that the chairwoman of the International Committee for Contraception Research, Dr. Regine Citric-Ware, is here. Shelley founded the ICCR in 1970, and it is still going and thriving today. Under Regine's very able leadership. And he would have been honored and I think very humbled by the fact that there was a committee of people who got together to raise funds for this event and for this lecture series. When you do something like this, you have to raise funds in order to pay for all the things that make it happen. And we have 3 members of the committee here tonight that helped to do that and undertook that work. And he would be very pleased that Jerry Weissman, Bill Speck, and David Madden are all here tonight, representing the committee. Shelley would be especially delighted to see our guest speaker, Dr. Alan Guttmacher, who will be introduced in a moment. We could not have a more fitting tribute to Shelley than to have a scientific lecture delivered by someone who is so dedicated to promoting research to address the most pressing reproductive health needs of women, men, and families around the world. And this is the kind of event that Shelley loved almost beyond any other. We'll have a Q&A at the end of this, and I could guarantee you that if Shelley was here, he might not have asked the first question, but I bet he would have asked the best question here. Last but not least, Shelley was always the happiest, and I always see him with his biggest smile when he was surrounded by his family. And we're so pleased that so many of his family have joined us. Harriet Segal, Amy Segal, Jennifer Madden, Andy Blum, and Dave Madden are all here from the family, and we're so pleased to have you here tonight. Shelley was an important leader of the Contraceptive Research and Development Program at the Pop Council. Under his guidance, we developed highly effective, long-acting, reversible contraceptives, Norplant, Jadel, Mirena, the Copper T IUD, and the list goes on. And even with those achievements and many others, it's difficult for me to capture in words the full range of Shelley's personal characteristics and his impact on our field and in the lives of individuals all around the world. He was a decorated scientist, a devoted mentor, a world-renowned humanitarian, and a crusader for health and rights of women, men, girls, and boys everywhere. And he was also a great friend to many people. The warmth and depth of Shelley's friendship knew no boundaries due to age, social status, geographic location, or other factors that often deter some of the rest of us from making meaningful connections with people in our world. Shelley was very special in that way. And now it's my pleasure to introduce someone for whom Shelley had a deep and abiding respect and friendship, Peter Donaldson. Peter has worked in the population field since 1973 and has served as president of the Population Council for the last 10 years. And Peter and Shelley have a lot of things in common. Like Shelley, Peter embodies a deep commitment to improving the lives of the most vulnerable populations, wherever they may be, an enduring belief in the Population Council's mission, and a profound appreciation for disciplines that extend beyond his own, in Peter's case, demography. Under Peter's leadership over the last 10 years, the Pop Council has grown in size and in prominence and in influence. Peter's retiring as president at the end of this year, and I know that Shelley would have been very pleased and proud to have Peter here to introduce our esteemed guest speaker at the second annual Sheldon Segal Lecture. Please join me in welcoming Peter Donaldson.
Thank you very much. It's very flattering to be compared to Shelley Segal, even if it's not true. It's so nice to hear. I want to add my welcome to Jim's and say a warm welcome to all of you here, especially the Segal family and our colleagues from the Marine Biological Laboratory. As Jim said, tonight is really a wonderful night for the Population Council. We're privileged to welcome one of the great scientific leaders of a global institution that is significant in its influence, its accomplishments, its importance for the global scientific community. And it's nice to be doing so on a campus like this, where there's been the professional home of 24 Nobel laureates, and it's been the home of the Council's biomedical research labs for the past 60 years, started nearby in the basement of Founders Hall. No one better represents the history and accomplishments of the Population Council than Sheldon Segal. As Jim said, I mean, under his leadership, the Council developed the most valued, widely used, long-acting, highly effective reversible contraceptives. And together they have averted literally millions of unwanted births and pregnancies. And they saved hundreds of thousands of women's lives. It's a tremendous record, and it's a pleasure to honor Shelley tonight. Jim said I started my career in 1973, and when I joined the Population Council in 1973, Shelley was already an important leader in the field, already vice president of the council, and already one of the champions of increasing voluntary family planning and improving reproductive health. One important thing I think that Shelley would be very proud of, and that we had an opportunity to show to Dr. Guttmacher early today, is our pipeline of product development continues to be very strong. And the next product that we're hoping to bring to market is a investigational contraceptive, one-year contraceptive vaginal ring. I think we're just a year or so from filing the FDA and getting the necessary approvals and so forth, so we can start to disseminate that method. What's interesting about the contraceptive vaginal ring is it's built on a technology that the diffusion of hormones over silastic that Shelley first identified and put into practice with Norplant and later Jadel. So that his impact is still very much felt. And we're working not only on new contraceptive technology, but on an anti-HIV agent that a woman could use to reduce the risk of HIV, and we hope that we feel like we're making significant progress and to have a new product available. Tonight, we're focused on our work on biomedical research, and that's proper given Shelley's history and Dr. Guttmacher's background. But I want to take a moment to call attention to a large body of research we have underway on programs that address the special needs of young girls. Among the places where we are carrying out that research is northern Nigeria, where those 276 girls were kidnapped. And one of the projects in our Abuja office is a project to provide safe spaces to girls the Nigerian government, Nigerian partners called off-track. And girls that are off-track are girls that are out of school or living without parental supervision or maybe in inappropriate work of some kind. And our effort is to provide a safe space where they can gain a sense of autonomy and learn some new skills and have some protection of being in a group. And we and the Nigerians hope that they can advance to a better life. And we're studying the impact of those safe spaces with the same kind of rigor that we study contraceptive safety and effectiveness here at CBR. So we want to, how can we make these safe spaces better and increase their effectiveness? This work is funded by the British government, by the British Aid Agency. And I recently received an email, late last week I received an email message from the British government official, the program officer who's responsible for this award, for monitoring this award. And he says, and I would say British understatement, recent events here underlie the seriousness of importance of this work. Indeed they do. So I think tonight I'm thinking not only of Dr. Guttmacher and Sheldon Segal, I'm thinking of those girls in Nigeria. And I think the reason we honor Shelley and the reason we're so delighted to welcome Dr. Guttmacher is because they understand the connections among the various streams of the Council's work. And they value and appreciate the seriousness and importance of the work that we have underway in Abuja. That was part of the great contribution that Shelley made to the Population Council. Tonight we're very pleased and proud to welcome an important public health figure, someone who has devoted his life to science and to the public interest. Dr. Guttmacher is director of the Eunice Kennedy Shriver National Institute of Child Health and Human Development, known by almost everyone under the acronym of NICHD. NICHD is one of the 21 institutes that together make up the National Institute of Health. The mission of NICHD, which was founded in 1962, is to ensure that every person is born healthy and wanted, that all children have a chance to fulfill their potential to live healthy and productive lives, free from disease and disability. And what strikes me about this is how close it is to the Population Council's mission. You remember the words of our founder, John D. Rockefeller. He said one time, the only reason for concerning ourselves about population is to help make it possible for individuals everywhere to develop to their full potential. So given this shared mission, it's no surprise that NIH and NICHD and the Population Council have been important partners for a very long time. And we've received lots of generous research support from NICHD, which really has enabled us to carry out all of the fine work that we speak about. They've helped us with our basic biomedical research, our research on new fertility regulation compounds. They helped us a lot with the clinical trial for our contraceptive vaginal ring, and they've supported path-breaking social science research at the Council. So we are deeply grateful for this support and exceptionally grateful for this long-standing partnership. Dr. Guttmacher is a pediatrician and medical geneticist. He was trained at Harvard Medical School and did his internship and residency at Children's Hospital. And after leaving Children's Hospital, he joined the University of Vermont College of Medicine and was instrumental both in their clinical and research programs there. He was named director of NICHD in 2010, but he had spent a decade before that at NIH, at the National Human Genome Research Institute, where he oversaw efforts to both advance genomic research and to integrate it into clinical practice and healthcare. Dr. Guttmacher has also played a very important role in reproductive health institutions. He was chair of the Guttmacher Institute Board of Directors or Trustees. He was chair of the board of the Planned Parenthood Federation of Northern New England, and he was a member of the board of the National Planned Parenthood Federation. And all these organizations, as you know well, are promoting reproductive health and family planning. I have to say for myself, I've been particularly impressed with Dr. Guttmacher's contribution to preventive medicine. Several years ago, he had a serious heart attack. It took place at home, and his wife turns out to be a certified EMT. And she began cardiac pulmonary massage immediately. And his takeaway from this experience was reported in the New England Journal of Medicine was never sleep with someone who doesn't know CPR. You might think that such a distinguished professional, someone who oversees the distribution literally of billions of dollars of research support and who comes from such a respected lineage, would be unapproachable and aloof. And I want to read you the email message. I sent him an email message saying, please come and speak and deliver the second annual Sheldon Segal Lecture. And this is the reply that he sent. He said, somewhere, either on my birth certificate or my NACHD contract, or both, it says I cannot decline an invitation like that from the Pop Council. And that it is named for Sheldon Segal puts it way over the top. Definitely sign me up. So, ladies and gentlemen, we're very fortunate to have a friend of Sheldon Segal's, a friend of the population councils, a friend of the global scientific community, and most important, a friend of women and their families around the world. So please join me in welcoming Dr. Alan E. Guttmacher.
Thank you very much for that very kind introduction, Peter. It's true that if the Population Council had asked me to do anything, I would have just immediately signed up to do it. But the fact that this lecture is named after Sheldon Segal really did, put it in a different stratosphere kind of thing. So it's an incredible honor and pleasure to be here to talk with you all tonight. And I'm very aware in doing that that those of us who labor in the science of all sorts, but certainly in science that touches upon reproductive health and well-being, that it carries on a wonderful tradition of science and medicine, the sort of laying on of hands from the previous generation, that we clearly, the work we do today is derived, as you've already heard. We're doing things today that are still using the kind of approaches that Sheldon Segal was instrumental in developing. That we clearly do stand on the shoulder of a whole host of pioneers. I couldn't help, and several of you have come up to me before this talk and mentioned this fellow too. I'm named after my Uncle Alan, my father's identical twin brother, who actually is also the obstetrician who delivered me. So my birth certificate is signed by Alan Guttmacher, which I think shows an incredible level of precociousness on my part. But as many of you know, he was, for many years, the head of Planned Parenthood Federation of America. And both he and Shelley, besides sharing moments together and science together, they were of a generation that was remarkable. In some ways, they were of two different generations chronologically, but they were both active in overlapping periods. And it's a period in which both the science and the social construct of contraception was so radically changing and developing. So while each generation will add more to that, this really is the founding generation. These folks, not just the two of them, but the people with whom they worked in many different ways, we owe just an incredible debt to. And I'd like to think that if they were here today, noticing the state of affairs, they would both comment that it's wonderful. Of course, as you've already heard, that contraception is about options, and that it is remarkable and wonderful that we have more options available today, clearly, than when they started their careers. But I think, knowing my uncle quite well and Sheldon Segal a bit, that they would say, that's wonderful, but so what? That's great that we're doing better than we used to, but we're still not doing well enough. There are incredible unmet needs, even in the US, but certainly internationally. And both, because they were so devoted to the science of this, would say, that's great. What are the new scientific opportunities? Tell me about those. What are they? And once you told me about them, get to work doing something with them. I'd be interested to hear about them, but I'd be even more interested in seeing them being developed and put to use. So what I'm going to talk about is a little bit about where we were back in their day, perhaps, and where we are now in terms of contraception. Briefly about where do we need to go, though I think most people in this room know that, so I'll be pretty brief. And then talk a little bit about whether this special, in some ways, new science of genomics, might that be one tool in helping us get to where we need to go. Talking about how genomics can be a tool to help us understand the biology of us humans in a better way. And also that it can allow us to do what I'm titling here agnostic drug discovery. Instead of doing very targeted, we know this thing does this, so let's come up with a drug to affect it, in a much sort of more general, as you'll see, agnostic way to think about the way our bodies work and how we might interfere with that in some ways. And then also bring up a kind of tension here, I think, that genomics presents between individualized, the healthcare, and public health. And then say something very briefly at the end about genomics is an interesting sort of model and influence in the culture of science that I think is important for science in general, but specifically for contraception science. So in terms of where we were, where we are now, I thought, well, first of all, it is important that science and medicine exist in a cultural context, and I know there are folks in this room who are old enough to remember, because some of them were just coming and speaking to me before the talk about the era in which, it's easy to forget that birth control in the US, contraception, wasn't even legal for married couples at one point in most of the US. And during the 60s into the 70s, that obviously this changed, but that societal change is incredibly important in terms of reproductive health and rights, which I think are inextricably tied together. But because they are tied together, we need progress in each to further the other. But I thought, well, let's go back. What did they know about contraception in 1962? So I thought I'd ask an expert. Well, I'd ask my uncle. He wrote this book, Pregnancy and Birth. I still have rather older women who come up to me and say, you wrote this wonderful book that got me through my pregnancy. I actually had this when I was a 20-year-old college student. A woman came up to me and said, I can't thank you enough. And I'm thinking, lady, what are you thinking? Anyway, Uncle Alan wrote this book, which really was, in those days, the only book for the lay public about pregnancy. He started writing this book in the 1930s, and it says something, I think, about his pioneering spirit, similar to Sheldon Segal's, that, in fact, this was frowned upon in academia. To be writing a book for the lay public, you not only didn't get any credit for it, but it was kind of held against you. But he did it anyway because he thought it was important for women to understand about their bodies and pregnancy. So he lists in 1962 edition of this book what's available in contraception. Well, number one, these are his exact comments, some words taken out, but all of the words here were his. The birth control pill, the most trustworthy method yet devised. Number two, the condom, ideal for some couples. It's interesting to go through these to think how much the world has changed, but in some ways how it hasn't changed. Number three, the diaphragm, properly used, as an excellent method. Number four, aerated contraceptive cream, simple and quite reliable. Number five, other chemical methods such as non-aerated creams and jellies, tablets, and suppositories, probably less reliable. This next one, I think, is someone that's my favorite. Coitus interruptus. There was a misprint in this. Now, that's almost the Latin for repeated acts of... So I can see why it would be relatively inefficient as a contraceptive method, though I'm not sure about the undesirable part. I assume in the next edition, I meant to check this week that typo was picked up. But for any of you who have the 62 edition, take a look. It's page 238, I believe. And then he talks about the rhythm method. Rather than rating it, he goes to give several paragraph how-to kind of sort of frowning discussion of it. So that's where we were in 1962. Now, this is a sort of table from various sources about, in the US, women's use of contraception. Now, for the first several periods of the survey, it was of married women only, because, of course, it was not appropriate to be asking unmarried women about such things if you were part of the federal government. Now, luckily, we're able to ask that question. So for the last one, it's of all women, not just married women. And this breaks similar data down by different forms of contraception, not all forms listed, obviously, but there are 5 listed here. And you can see, again, how trends change over time. And again, the last 2 survey points are of not just married women, 15 to 44, but of all women in that age group. And this is looking specifically at the most recent data, really 2006 to 2010, and looking at the contraceptive methods used by, again, all women age 15 to 44. And it's just an interesting state of where we are. You'll see that oral contraception and female sterilization both are quite large. This is looking at developing countries. In 2012 from the Guttmacher Institute, again, a reflection of how the The international scene is in some ways similar, but in some ways distinctly different from the US. And of course, the international scene is distinctly different depending upon what country and even what region within what country one looks at. This is looking at the prevalence of usage in 2000 for women in a union, whether marriage or some other form of union, ages 15 to 49. So if that's where we are, where do we need to go? And it's not telling folks at the Population Council anything they don't know because they are the vanguard of achieving this, and that's that we really need to develop more and better options. We need male agents beyond the condom. We really do need male agents, and again, the Population Council is leading agent in terms of trying to find those agents. Female non-hormonal agents, longer and short-acting agents. We need, again, to have options, reversible, non-reversible agents, dual purpose, really multi-purpose agents, dual purpose in terms of being both effective in contraception, but also effective in terms of infectious disease prevention. And then we clearly need, and I think this is something that some of us have been late to coming to. There was an idea, I think, for a long time that there would be a silver bullet, a magic contraceptive agent that would work for everybody all the time, et cetera. And that's not the way people live their lives. Even for a specific individual or couple, the ideal contraceptive will vary depending upon where they are in their life cycle, where they are in their life stages. And we need to develop better options for all of these different settings. Now, the good news is, and you could add lots of other things to this table, and this comes from good folks here at the Pop Council, from the Bill and Melinda Gates Foundation, and ICHG, and lots of others, that there are lots of technologies that are in the research and development pipeline, which is wonderful, much larger list than we would have ever had before. On top of that, though, it's easy to focus on contraceptive technology development, incredibly important. But we also, and again, the Pop Council plays an important role in this, we also need to understand contracepting behavior much better than we do. We need to understand the basis for people's choice of contraceptive options, for those factors that influence use to make it more or less effective. What are the factors that lead to discontinuation of use? We really also need to think about ways of reinventing the drug and device development pipeline. We've had sort of monolithic models of that largely have come out of pharma, that for multiple reasons, and again, the Pop Council has been creative in invention here, that for contraceptive agents particularly, we need to think of other steps than the traditional both drug and development pipeline in order to make things work. And then we need much, as some of us were discussing earlier today, more and better implementation science. It used to be that wonderful non-governmental organizations, NGOs that were out in the field, they looked at the results of what they did. That is, how many contraceptive agents did we hand out? And that was the scorecard. Well, that's wonderful, but you need to know, in fact, is it having the desired impact? Are women and couples being able to control their reproductive lives? That's not simply the question of whether they have an agent. Is it working? Is it having side effects? What are the other kind of effects of it, et cetera? We need to have richer science in that space as well. So can genomics help us in terms of answering any of these needs? And I hope that I'm going to convince you that at least for some of this, I think it can be helpful. But we take a step back first and give sort of maybe a primer in genetics, genomics. This all comes out of the Human Genome Project. This was a project launched in 1990 with the ultimate goal of sequencing the human genome, this genome that we all share in common. And it's interesting just in terms of its context, it really sort of started the study of modern genetics about the same time as modern contraception development began in the 1950s. And it was 50 years to the month from the April 1953 publication by Watson and Crick of their model of the DNA double helix, which underlies our genome. Exactly 50 years to the month later, that we had achieved the sequence of the human genome, the Human Genome Project, ending in 2003. I can tell you as someone who was involved in the tail stages of this, I was the deputy director of the Genome Institute, which was the lead institute for it at the end here. And we thought it was kind of important. It just wasn't good PR somehow. We would feel bad if it took like 50 years plus 3 months. You know, we really needed, and it was a wonderful thing to whip all the folks working on it into line. You know, you've really got to produce. We've got this April, you know, deadline. We made it with 2 weeks to spare, so we were very happy about that, that we really did sequence this human genome. Now, why do people still talk about the Human Genome Project? Well, it was your typical international government project, like all of those ends ahead of schedule, and like any ends under budget. Whenever we go to Capitol Hill, we somehow mention both of those factors. It was also interesting in that it devoted 3 to 5% of all of its funding to looking at the so-called ethical, legal, and social implications of the science, which 3 to 5% may not sound like that much, but it made it far and away the largest funding of biomedical ethical thinking ever. And it was really important for the field, and it is colored, and I think still is very much a part of the field of genomics. Understanding that we are letting a huge genie out of the bottle with this, and that we'd better think about the societal implications of this, not just the scientists, but policy leaders, society in general needs to be thinking about these things before the genie is unleashed rather than after the fact. The Human Genome Project also did some other things. It showed that centrally coordinated big science has much to contribute in biology. Before this, the only model really for biological was the individual investigator, sort of Madame Curie over a flask in the lab, still is the bedrock of scientific research. But there are certain situations where a kind of coordinated top-down approach can be helpful, and I think the Human Genome Project was a good example. It also proved that non-hypothesis-driven research can be important. A lot of people in science think you have to have a hypothesis, otherwise why do the experiment? Well, there was no hypothesis to the Human Genome Project, unless I guess the hypothesis was we hypothesized that we can sequence the human genome, even though we have no idea how to do it. There really wasn't a hypothesis. We just thought, gee, if we can do this, it will enable all kinds of other hypothesis-driven, investigator-initiated research later on, which is what did happen. But I think its greatest impact, and it's not the Genome Project alone, it is society in general. The Genome Project, so sequencing the genome, every night it uploaded all of the information onto a computer, and anybody else in the world who had a computer and paid their electricity bill could download that information every morning. So every 24 hours, all of the data was made completely publicly available, which incredibly accelerates science. We have had in science, I think, for a long time, this sort of idea that the data belong to the principal investigator. And we need to come up with ways to recognize and reward not just the principal investigator, but all members of the research team. But in fact, I would argue that scientific data, particularly if it's paid for with taxpayers' dollars, belongs to society. And we need to come up with ways to make that data as widely available, as quickly as possible, because that's what accelerates science. That's what accelerates the benefit of science to humanity. And that, again, I think is an important, to me, the most important aspect of the Human Genome Project. It also gave us some interesting facts. So, such as how many genes do we humans have? So this is a multiple choice. You have 2 multiple choice questions. And depending upon how you do with these determines whether you get dinner later. So here's some context. I want you to guess how many genes do we humans have. So influenza has 1,700 genes. E. coli, a bacterium that you hope is not in your dinner, has 4,300 genes. The fruit fly, Drosophila, a mainstay of biological research, has about 13,000. The lowly worm has about 18,000. And a animal species, Arabidopsis, or cress, mustard grass is sometimes thought, has about 25,000 genes. So knowing that, I'd like you to guess about how many genes do we humans have. Now, the fact is that you have 20,000. You have less than a plant. So I hope that gives you pause. There is a wonderful picture of Arabidopsis. And over dinner, I would suggest you might want to contemplate the cosmic significance of the fact that you have fewer genes than a plant. And I hope you will treat your salad at dinner with newfound respect. So it's clear it ain't the number of, because I would argue we're somewhat of a more complex organism than a Theocress. So it's not just how many genes you have, it's how you use them that really makes the difference. Now, I think the other thing that's very important about the Human Genome Project is that we showed that we are all mutants. I am here from the federal government to tell you, you are a mutant. We're all mutants. Nobody has a quote-unquote normal genome. We all have hundreds of variations, probably scores of which are not so good for us, that make us who we are. And many of them have to do with disease process. Some of them have to do with other characteristics, intelligence, other things like that that are not determined by your genes but heavily influenced by them. Again, none of us have normal genomes. It's a whole different lecture. I'll be happy to talk about that in the question and answer period, perhaps. So let's talk about some real-world applications of this whole era of genomics. You know, we've had the human genome sequence for over a decade now, so where's it going? Well, for one thing, it's important to realize that I would argue that all 10 of the leading causes of mortality have genetic components. This is a list for the US in 2010. Interestingly, for the developing world, the list is beginning to look more and more like our list. Now, the only one there that some people would say, well, gee, unintentional injury, what does that have to do with their genome? It's unintentional injury. Well, first of all, there's a genetic factor that we've known for years, characterized a long time ago, that actually has a heavy role in your risk for dying of unintentional injury. And as a clinician at heart, I can tell you my physical diagnosis skills are still good enough that I can look around this room and identify a number of people who have this genetic variant. Yes, you've done the same thing, this genetic variant that puts them at a much higher risk of dying of injury than other people in the room. And without your informed consent, I'm about to tell you, point out to you which one of you have this risk factor. The risk factor is something called a Y chromosome, which makes you a male. And as my wife points out to me much more frequently, I think I need to be reminded, it may do nothing useful for you, and it does make you much more likely to die of injury. Now, maybe that is simply a social construct, not a biological one. But if you think that's the case, please point out to me the society where that is not the case. Now, even if you forget the X and Y chromosome for a moment, there are other genetic factors that have to do, for instance, with neurotransmitters that may have some impact upon one's aversion or welcoming of risky behaviors, for instance. So it's actually plausible that well beyond that there are other, and clearly if you suffer an unintentional injury, the chance of actually showing up this table because you have died from it clearly depends on your genome, because how we as biological organisms react to that injury or any illness. People focus on the genome, your likelihood of developing illness, very important, but just as important in terms of how you are going to deal with that illness once you develop it. So there's a cultural impact. The genome has been very good for magazine sales. There have been, I could probably do an hour talk simply on various magazine covers that are shown genomics. That's a relatively recent one from Time magazine. Personalized medicine, I don't want to single out Duke. Almost any medical center worth its stripes is now having a personalized medicine clinic, program, initiative, whatever, because the idea that knowing your genetic makeup, your individualized, will really enable us to get to an era of personalized medicine. I don't mean to poo-poo that. I don't think we're quite there yet, but we will be there where, in fact, your individual genetic makeup has a huge impact on how you metabolize drugs that we prescribe for you, in your likelihood for developing various diseases, et cetera, and we're just in the era where we're beginning to understand those things in a way that I'll show you in a moment. You can do personalized genomics. There's a company called 23andMe. There are a number of them out on the internet where you can swab the inside of your cheek, send in your DNA, get various information back. You can get less information back from 23andMe than you could a few months ago because the FDA has done a cease and desist order on some of the things they were sharing back with people. So there's thorny issues there. You could argue this is a editorial or a cartoon that came out in 2001 when we did the draft sequence of the human genome. And it's a woman and her, I guess, boyfriend sitting at a counter. And she has, you can see tucked away in her purse, the Human Genome Report, presumably his, because he's holding out the engagement ring and she's saying, let's just be friends. And he's saying, it's my DNA, isn't it? So obviously just the, nice story, how could genes have to do with your likelihood for marriage. Well, this is an actual website, Dating DNA. Now, they claim that you could swab the inside of your cheeks, and they would tell you whether you were compatible, with whoever your potential love interest was. Now, I can't tell you whether that is still up or not. I got this screenshot 10 years ago. I've been curious to go back and look again, but literally my government computer will not let me access the site. And at home, my wife won't let me access the site. So I wish one of you would go check it out and let me know whether it still exists. I think that it got a little bit beyond the science here. Hucksters occupied the space. But there's real scientific impact, the whole area of so-called epigenomics. Interestingly, it turns out that, it's neither nurture or nature, it is both. But in fact, in some ways, your environment, using that term quite broadly, not just toxic smokestacks, but including that, but your social environment, everything about your environment, actually not only interacts with your genome to make your life what it is, but it modifies your genome. And we're beginning to understand more and more about this, that very early in life, in fact, even in utero, that there are adaptations that the environment forces on your genome that you carry around with you for the rest of your life. So the idea that the child is father of the man, and I would say of the woman too, we're beginning to have the science so-called developmental origins of health and disease, that very early life. And in fact, we're beginning to get some data to show that these epigenomic changes hold true over at least several generations. So not just your experience in utero, but your mother's experience in utero may impact your lifelong health. We're just beginning to understand the science of this. It's important that I say that we are not in the post-genome era. It was interesting, we did this Human Genome project and everything. There are a whole bunch of editorial comments that came on. It officially finished on April the 14th, 2003. And there were a bunch of editorial things about we have now entered the post-genome era. These all appeared on April the 15th. Well, all we knew about genome eras was that we were in the pre-genome era through April the 13th. And if we were suddenly in the post-genome era on April the 15th, it meant that the genome era itself was April the 14th, 2003. If you slept late, if you went to the beach or something, you completely missed an era in science. We're not. We're at the very early stages of this. So the applications, and it's just like most new technologies, in some ways it just is a technology. I think people at first way overestimated its immediate impact, but they tended to underestimate its long-term impact. The same was true of the telegraph, computers, any other new technology. So we're just really at the beginning of the genome era, of having our genome and being able to harness it. So I thought I'd give you one fairly graphic representation of this. This is a series of scientific research called genome-wide association studies. The basic idea behind these was, and it was really founded on this graph. This is a graph that appeared in the journal Science in 2002, and it showed what did we know then about genes in human disease. And the good news was here in the pink, because you get to use the scale over here, there were about 1,700 different traits, most of them diseases, for which we knew the gene involved. Now, those are so-called Mendelian traits. It means that basically if you have the genetic variant or mutation, you will have the disease. Things like cystic fibrosis, sickle cell anemia, other kinds of diseases. So it sounded like we knew a lot. In fact, not so much when you realize that there's 6,000 or 7,000 such diseases in the world. It's only a third of them that we know the genes involved. Where it really got worse was when you looked at complex traits. What does that mean? That means where there are multiple genes and the environment involved. So much more common things like heart disease, like schizophrenia, like diabetes. So for those, unfortunately, I have to use the scale over here, so we're only down around 30, but in fact, you'll notice it doesn't say human in that. That's for all species, mice, rats, monkeys, whatever. For humans, we were down here in single digits. There are hundreds of these common disorders that we can get, but we only knew genes involved and not most of the genes involved in less than 10 of them. So that wasn't very good. So we thought, our problem is that we've always done sort of targeted genetic testing. Basically, we think we know something about the biology of disease, so we say, well, gee, high blood pressure, calcium metabolism is involved in that. So we'll look at calcium genes. So we'll take 1,000 people who have high blood pressure, 1,000 people who don't. We'll look at genes that we know have to do with calcium metabolism, and we'll see whether the genetic makeup of the 1,000 with the disease looks different than the 1,000 without. The problem is that our bodies are way too complex for us to be able to figure this out. So we said, let's do a genome-wide association studies instead. We've about to sequence the human genome in 2002. We have that available. So why don't we agnostically look across the human genome? We'll sequence 1,000 people with the disease, 1,000 without. We'll sequence their whole genomes, see where the genomes vary, and then we'll focus in there to find the genes that are involved. So that sounded like a pretty good idea. We did the calculation in 2002, and would only have cost $10 billion to do that for a single disease. So if you heard, there are 21 institutes at the NIH. So we thought at the Genome Institute, we thought, you know what we could do? At that point, the NIH budget was around maybe $24 billion a year. We'll go around to all the other institutes, and we'll say, look, we'll pick one disease, and you give us about half your budget for the year, and we will figure this out for the one. And there's at least probably a 5 or 10% chance that it's actually a disease of interest to your institute. Wouldn't you like to do that? Well, we figured that was a bad idea, so the envelope in which we did the back of the envelope calculation went in a desk drawer, never to be brought out again. Well, not quite, because what happened is that the cost of sequencing, this is Moore's law. In computing, it said that every 2 years, the price of computation goes down by 50%, which is pretty good. I mean, this is, whatever we called that decade, the aughts or whatever. The only thing falling faster than that was your 401k during that period. So that's pretty good. And we kept saying in the genome world, the genome sequencing is following Moore's law. In fact, it was doing a little bit better than Moore's law, and then you'll see it started doing way better. So the price of sequencing the genome went down so drastically that by 2007, that same study that in 2000 was going to cost $10 billion had gone down to $800,000. And today, the genomic sequencing for this would only be less than $80,000. So we thought, gee, this might be a way to identify genes that play roles in specific common disease, which would then allow us to say, gee, you have a variant in this gene, you're at increased risk for this disease. But also, and this is where I think it becomes particularly important in terms of contraception, allowing us to understand the biology of human life in a way that I'll show you in a moment. So this is just this visual of, these are looking at genome-wide associations. The first was done in 2005 for what's called adult or age-related macular degeneration, the first or second leading cause of severe vision loss in our society. So that's nice that we had, now we went from maybe, remember that earlier graph, we went from maybe 8 to 9 disorders for which we knew a gene involved. In 2006, several more showed up. In 2007, it began to happen so quickly that the artist doing this was doing it every quarter of the year for us, and you can see the rapidity which this happened. So in fact, it happened so frequently, by the end of 2010, the poor guy at the NIH was making this diagram for us basically said, either you let me stop making that diagram or I am quitting. And he was a really good artist, so we said, all right, you don't have to keep up, because he had to keep changing the font size and everything else because there was just too much science. So he said, do one last one for us a couple years later. So he does this thing, which is showing there are now hundreds of genes implicated for a wide variety of different kinds of disorders. That's the color coding here. And so if you did this same graph now, we're off the chart, literally. We've gone from a handful to hundreds of genes implicated in common human diseases, a huge change. Now let me go back to thinking about how this might be these kind of genomic tools might be some use to us in terms of thinking of contraception. So the first thing is explicating biology. And let me go back to the first, as I mentioned, the first one of these done in 2005 was done about age-related macular degeneration. And if you looked at that diagram very carefully, you would have seen eventually there were 3 different genes involved with age-related macular degeneration that were shown to have a huge contribution. They contribute over 50% of individuals' risk for developing AMD. Well, what all 3 of them had in common is they were involved in pathways having to do with inflammation. Now, before this was done, nobody, except for apparently a very small group at Ohio State, nobody thought that AMD was an inflammatory process. It was thought to be due to neovascularization, the formation of new blood vessels in the eye, or simply senescence. Your eye got so old it didn't know how to do things right anymore or something. But in fact, the fact that all three genes involved in this had something to do with inflammation made us think, aha, maybe this is an inflammatory process. Well, we have categories of drugs called anti-inflammatories. Maybe they would be of use for treating, or even potentially for the many people at increased risk of developing the disease, for preventing the disease. So there are now trials going on to see that. But what we've seen for disease after disease, as these kinds of things become apparent, is for disease after disease, it shows us the genes involved turn out to be in pathways we never suspected. So they tell us something about the biology, not only of disease, but of health in ways we never knew before. So they give us whole new categories of targets because we understand the biology of disease processes or health processes, like fertility, in ways we hadn't before. So if you discover genes that are involved in fertility that you never knew about before, completely new potential targets to focus on. Now, the idea, so other things, for instance, Crohn's disease, common disease, where again, having these genome-wide association studies has allowed us to identify multiple genes involved in Crohn's disease that we never knew about before, developing new animal models to replicate the effects of these genetic variations, again, in terms of contraception. If we can develop genes that we've never thought about in terms of having an effect on, in terms of fertility, that we can develop animal models that overexpress or underexpress these genes so that we can do animal modeling to be much more effective for us. We can do so-called chemical screens to identify new candidate therapies. What does that refer to? Well, that's this idea of this sort of agnostic drug discovery. So here's your second multiple choice quiz. And this one I've actually really labeled the choices so you have to declare yourself. You now know, if you didn't before, that we have about 20,000 genes. I would remind you, less than mustard grass. But we have about 20,000 genes. So if we take all of the drugs that we have available to us, the over-the-counter drugs, the prescription drugs, the street corner drugs, the under-the-counter drugs, all the drugs known to us, how many of the human genes and their products, the proteins they make, do you think we have at least one drug targeting? So we might have all 20,000 genes, 100% of the genome is, we have some drug that targets. So make up your choice. If you're really gutsy, you can tell the person sitting next to you what you know the right answer to be. But the right answer is 500. Only 2.5% of our genome do we have a single drug targeting. Now, it may be that much of our genome, as that's said, is not druggable. That it just makes proteins that we can't somehow inhibit or do something with. That's probably true. We obviously don't know yet what percentage. Let's argue that half of our genome is not druggable. Well, that's great. That means we've occupied 5% of biological space instead of 2.5%. We still have lots of potential here. And this is a question, again, I think for contraception, very important. It's not just tweaking known drugs so that they're slightly more effective or so you might take it every other day instead of every day or something. This is completely new categories that are conceivable, and there are various sort of high throughput ways of screening for these now. One example from a very different world, again, is for a rare disease called Niemann-Pick disease type C1. It's so rare, not a single drug that's effective against it. And using this sort of chemical genomics approach, a drug that's already out there that we had no idea had an impact on this disease process, within a year of that discovery was already into FDA-approved trials. The human microbiome is very interesting. After the Human Genome Project was done, started looking around for what else can we sequence now that the price is getting cheaper and cheaper. What about all those microbes we carry around with us? So I'm here from the federal government not only to tell you you are a mutant, but also to tell you that most of the DNA you have with you at this moment is not yours. Now, it says nothing about your personal hygiene or other practices. It has to do with the fact that we have an unbelievable manifest of bacteria that live in and on us. And many of them are incredibly important to maintaining health. But some of them are not so great, and they can cause disease. So, for instance, we've already done, you'll see here, urogenital was one of the 5 human sites where we first started studying the microbiome. Besides discovering all kinds of organisms we never knew existed before, understanding how various things change that normal flora is very important. For instance, one of the things that we at NICHD are very interested in is there is a fundamental difference in terms of being born vaginally or being born by cesarean section. And that fundamental difference is if you come out through the birth canal, you are colonized by the bacteria in the birth canal. That's the way we human organisms were designed to be born for eons. Recently, a larger and larger percentage of kids are born by cesarean section, and they don't get the benefit of that bacterial inoculation. Does this deprive them of something? We're beginning to have some data that says maybe it does. But when we begin to think about particularly intrauterine devices, if we fail to take into account a woman's microbiome, we may fail to realize why some agents are more effective in certain women than others, why some agents have side effects in some women and not others, and not for anything basically in or on the body. We're beginning to understand that. And might we be able, in fact, to have an impact on fertility simply by changing someone's microbiome? Is there some way of taking an oral, some kind of yogurt preparation that someday will change your fertility? So this gets in this question of individualized healthcare versus public health to me. We clearly are beginning to have information that individual genetic makeup definitely changes the way we metabolize drugs, which means that for some people, the desired effect of a drug just doesn't occur because they metabolize the drug to an inactive form so quickly nothing happens. Other people have a side effect of a drug because they metabolize it in some way that makes it a bad actor. Some people, it's not them, it's the microbiome of their GI tract if it's an orally taken drug that does that. Also, in terms of contraceptive behavior, really genes do have some role in behavior, ways that we're just beginning to understand. Might in some way genes have an impact on individuals' behaviors around contraception? Now, this brings up questions about, I think, both individual versus public health care, but also sort of population health. For geohistorical reasons, the propensity of a specific population to have some genetic variant can vary incredibly. Let me give you one example, having nothing to do with contraception, but this is a frequency of a genetic variant that's very important for metabolizing a drug that's frequently used for certain indications, and it can cause a very severe side effect called Stevens-Johnson syndrome, which basically can lead to sloughing of almost one's complete skin. And it turns out that there is a population variation in the frequency of this unusual, in general in the world, allele. In areas of the world shown in green, it's very infrequent. Only less than 1.6% of individuals have the variation. Here, anything up to 12.8% of the population has that variation. You wouldn't want to be using a drug that can cause your skin to slough off. If 1 of 8 people taking it, we're going to have that effect. Just one example of many of the so-called area of pharmacogenomics that I think we need to think about. So that becomes a question as we start developing more and more pharmaceutical agents. Do we need to think about some being more successful in certain population groups than others? How do we balance? Because even, in any country, it's not just a question of where your ancestors came from, but it's a question of who you are as an individual. Someday, all of our drugs, presumably, including perhaps contraceptive agents, will need to be individualized to our genetic makeup. How do we do that in a cheap and efficient way that can be used in a low-resource public health setting? Should we think about that at all, or should we never even think about it? It's a conversation that we in the reproductive health world need to be having more of. We're not ready to do that now, but we need to think forward to the day where it would be technically feasible to think, is it worth doing? If so, how? Or maybe not at all. So let me say something to end up, you'll be glad to know, to end up about genomics and the culture of science. Well, I think, what does genomics bring to the table? Well, I think one of the other things it does, again, is changing this predominant idea of data to the idea that it's so, it's in the age of wikis and everything else, that there ought to be open access to data. That I think genomics has been very useful model for demonstrating how to do transdisciplinary biomedical research, bring people from very different perspectives, clearly work that the Population Council has long done. And in academia, it's a challenge somehow. How do we reward and recognize people for doing this? If somebody in academia is author number 7 on a 20-authored paper, but has done important work, I can tell you, having been on a tenure promotion committee before in my life, they're lucky if that's not held against them when they come up for tenure, let alone get credit for it. But if it's really important work in the world of multidisciplinary team science, we need to come up with ways for doing that. Underscoring the need for interdisciplinary fluency. In today's science, because so much science does require multiple perspectives, it's good to have in-depth knowledge of one area, but have some fluency in some second and third scientific languages, not necessarily German or French anymore. But if you are someone who is, a hardcore chemist, to know some things about biology. If you're, like me, a medical geneticist, to know something about biocomputation and these other areas, particularly biocomputation, because it's so across the board. I think the other thing genomics has shown is a cool science that tends to attract the best and brightest, particularly young students. There was this revolution in genomics because we got all of these really bright-eyed, just creative people coming in the field. And the great thing about young scientists, of course, is they don't know yet what they can't do, so they go out and they do it. Us old fuddy-duds know you can't do it, so we don't even try. They go out and they do it. We need to keep reinventing ourselves to do that. So the question is being cool. What do you need to do to be cool and attract new researchers? So I thought my sort of idea of a cool person. Those who are old enough in the crowd will recognize James Dean, not potentially the model of a scientist, but certainly the attraction, the identification of cool. So in science, you need to have an important problem to be cool, I think. You need to have some interesting new scientific way forward. You need to have timely exposure of the young researcher or even an established researcher to your area of science. You need to have role models that a person would want to emulate. You need to have effective mentors who are not necessarily the same as the role models. They can be, but you need to have effective mentors, and often multiple mentors, and today often effective mentors in different areas of science. And you need to have financial support as well, which is where the NIH and others come in. So, is contraception research cool? Well, James Dean's wondering, and he certainly thinks, yeah. Boy, do we have an important problem. Everybody in this room knows that, but much of society knows that. It's hard to imagine a more important problem. And on top of important, in terms of being cool today, it's a global issue. And as any of us who have children, grandchildren, whatever know, global health is so cool these days. There's so many people interested. So we have arguably the most important global health problem. So it's hard to imagine that we don't do well in that. Scientific way forward, I think we've got some. You saw that long chart of all the things, the R&D pipeline. I've given you a glimmering of one of, I think, a number of new kind of scientific approaches that can help us come up with more of a scientific way forward. I think we need to take it upon ourselves. Some of us were talking about training opportunities earlier. We need to make sure that we have a chance to get young scientists exposed to this early in their careers before they have differentiated into one area of science or another, so they can see that this is really important and something to do. We need to give them the role models that they would want to emulate, and that's often through this timely exposure we do that. We need to provide effective mentors to them, and we clearly need to provide financial support to them, because we need to come up with the next generation of Shelley Segals. We need to continue the kind of work that he did. We need to move forward with the work, but we also need to have new generations that will take this forward, that will have the same compulsion he had to both advance science, but to also advance it in a way that touches people's lives, particularly their reproductive lives. The Pop Council has long been at the vanguard of that, still is at the vanguard of that. So again, it's a real pleasure and honor for me to be here to thank you for your work, thank you for attention, and if I didn't completely overgo my lengths of time, I think we have time for some questions, answers, whatever. So thank you. Questions, thoughts, or discussion, how come you didn't say anything about this? Or weren't you wrong about that? I love those questions. But any thoughts you might have?
Thank you so very much for this beautiful lecture. You addressed the aspect which is very difficult of the pharmacogenomic, and the fact that in contraception field we want to apply some new molecules to large population with various background and various genomes. And it's very difficult because we do not want to get contraception as a kind of medical disease where you have to do a screening before you get into the prescription. Yeah. And that is something we have seen, for example, in the thrombosis issue, in thrombophilia, in certain area of the globe. So I think it's really something where maybe the progress of science can, have something so quick, cheap that you can screen quickly, but it is...
Absolutely. I think someday, with your work, there'll be more and more oral contraceptive agents, for instance. I think someday when a woman comes into her clinic and says, I'm interested in oral contraceptive, besides the whole option, well, we have this agent that you can use with this frequency, this agent for that there will be a panel that will be done in a lab within the clinic that will look at that woman's genetic variants and maybe something, if it's going to be taken early, something about her microbiome potentially even, that will say, besides these agents, based upon your genetic makeup, I really worry about using this agent because you're more likely to develop hypertension due to it or something else. Whereas this one looks really completely safe. Not only safe for you, but you're going to be able to take a lower dose than other women because you have this variant that means you're going to metabolize it in this fashion that we recognize in 5% of the population that makes it more potent kind of thing. Now, that day is far off, but I think absolutely that will help. And again, the question becomes, how do we, in a public health way, do this but still recognize the individual variation in biology? I think someday we'll be able to do that. That day is still far off, of course. How far off, Peter asks. Well, I think it'll come in the U.S. before it does in sub-Saharan Africa, for instance. There are some, not in contraception, but there are some agents now before you are prescribed them that a genetic screen of just a couple specific genes is done before you prescribe them. There are very few, but there are a few today. I think it'll be a common part of our healthcare, probably in about 20 years or so, I would think. But increasingly common. It's not going to be that we don't have and then one day we do. I think it'll become increasingly common over the next couple of decades.
I work in the field of system dynamics, which is basically focused on decision making. Mm-hmm. And you have raised more questions than I can think about today.
That's full employment for you, and, yes.
How can you use the software developed by the man named Jay Forrester originally, who invented random access memory, before which IBM was a typewriter company? And he's still alive at about 98, 6 maybe. How do you make the decisions of where to spend your money, where to get your money, what to apply it to, and who to do it? Those are a lot of decisions to make to get the best results.
And I don't know how to make those decisions, and it was made official because the NIH hired about 2 months ago our first ever, I can't remember the title, but it's basically our czar for big data. That we realized for all of science how we deal with these huge, it's sort of a scientific truism these days. The challenge in science is no longer coming up with data. It's trying to interpret and analyze the masses of data that we suddenly have. And you're absolutely right. So that's why this whole area of biocomputation becomes so important. How do we even store the data, let alone analyze it, is a huge challenge for those of us funding research, et cetera. Hugely complicated. I think part of what we need to do, besides, for instance, sharing data, we need to share access to the analytic tools. What happens today too frequently in a lab is if somebody wants to figure out how to analyze large amounts of data, they send some poor postdoc off for a year or two to figure out how to do it, and then once they do, they have their own private way of analyzing the data, which is not necessarily replicable in the next lab anyway. We need to be funding more large ways of looking at large amounts of data to be able to make analysis possible, so different people can look at those data sets simultaneously from very different perspectives, asking different questions, or even asking the same question with different methodologies, so we can do that. So we know the challenge in front of us, but I don't think we've got good answers yet. You're absolutely right, T.
Great. Al, that was a terrific tribute to Shelley, to your uncle, and to science. You mentioned earlier that there were questions about bioethics and there were questions about legal and social. Could you give us a little hint, a few of the things that were of concern to people who were moving this field forward?
Yeah. I mean, there's so many. Everything from the question of access. Obviously, some of this stuff is pretty complicated. So how do you make it accessible to people who have very little health care, who don't have the educational background to understand some of this, et cetera, et cetera. So huge issues of access. The question of who knows what when. For instance, many of you will know that since then, again, another thing is really started in the 1960s, we've had in the US and much of the world so-called newborn screening, where newborns are screened, depending, it's a state-run program in the US, it depends what state you live in, but today most newborns are screened for about 29 different inherited disorders when they're first born. So that gives us a fair bit of information. Those 29 are selected because if it were not for the screening, by the time anybody made the diagnosis, there would have been irreparable harm done to that kid. And we have an intervention that can be applied during that preclinical time that would make a difference. Phenylketonuria is the sort of the poster child for this PKU. But there are a couple dozen of those. Well, within 10 years or so, we will have the technology available for about the same cost of that to sequence a newborn's entire genome. So, wow. So what do we do with that information? Because it will tell us about the risks for lots of diseases that don't even show up till adulthood. So who gets that information? Do their parents get it? Does the pediatrician get it? Do we wait until the kid is 18? Because maybe the kid is like many of us, I'd rather not know that about myself. So how do we handle those kinds of things? What about the fact that, I've just barely mentioned it, that just as genes have a role, they don't determine, but they have a role in our risk for, atherosclerotic heart disease or whatever, they also do play a role in intelligence. So what happens when we identify each gene is going to have a small contribution, but there's, 75 genes that have to do with human intelligence. And then a newborn, you could screen that newborn. Wouldn't be part of their health screening, I would argue, but a parent can get that done and send it off to some lab and find out what are their kids' intelligence proclivities. Well, I've got 3 kids. I can only afford to send 2 of them to college. Do I decide not to send the one who doesn't do so well in this genome IQ test? It's those kinds of questions. So I hope I've given you enough so you won't be able to sleep soundly tonight, because it keeps me up at night. And I have to say, the more people that are worrying about these things, we can share the responsibility so I can sleep better. So thank you for asking the question.
Other questions? Yes,
one of the issues is bringing the great technologies to the women who would use them and the men who would use them, and we're very dependent on the commercial pharmaceutical industry and others. There was an article this week about very little research is even going into antibiotics anymore, only chronic diseases. Maybe fertility is a chronic disease, I'm not sure, but how do you think about that issue and also the litigation risks and other kind of risks associated, which have really stymied innovation in this area?
There's no question contraception. I mean, many of you in the room are world experts on this. You know, one of the real challenges has been the lack of interest of pharma between litigation, between having difficulty sometimes, which either already other agents out there, why is this new one going to sell kind of thing, and also sometimes concern about various social boycotts and other kinds of things. So it's incredibly complicated. Again, the POP Council and ICHE and others have shown some innovative thinking. There's a big push at NIH about reinventing the drug discovery pipeline that's usually thought about in terms of just some of the traditional steps in pharma to make the whole process quicker and cheaper. But also, I think it brings up issues about coming up with ways of doing this, and Plan B and others have come up with ways to do that to avoid some of the potential stigma and other things that can be associated with reproductive health. I think we need to not copy old models, but continue to re-engineer the process, both in terms of the biological steps, but also in terms of some of the social context parts.
Yes, thank you very much for a wonderful talk. I've been thinking about how we take this tremendous opportunity for the future with what's been discovered so far and help young people in our educational system to harness this new information so they can utilize it in meaningful and productive ways.
So it's a great question. And I think the wonderful thing in the revolution in information is that obviously people find it easier and easier to get lots of information these days. But we need to utilize that in better ways. And there are ways I think you can marriage both research and, for instance, here's something that we're having a meeting on June the 30th to bring together a number of organizations interested in the following question, which has something to do with a different aspect of reproductive health. And that is, it's astounding that considering it's an experiment done 4 million times a year in the US alone, that we still don't really know the definition and the characteristics of normal human pregnancy. You'd think we would know that by now. So we're coming up with a kind of a way to have an interactive web-based presence, which has some problems, but these days less and less of a problem, a web-based presence where we will combine both research and information to people, we hope, by having women, inviting women to come to this nonprofit kind of thing and tell us about their pregnancies in a kind of structured, interactive way. For a woman who wants to do it every day, she's welcome to. A woman who wants to do it once a month can. Could you feel the fetus moving around today? Did you have any spotting? What did you have to eat today? When was the last time you saw your health care provider? What kind of exercise do you get? What kind of work do you do? But then also give information back. Gee, you're in week 16 of your pregnancy. By the way, how do you know that it's week 16, we might ask? But you're in week 16. Here are the things you might be wondering about. Here are some good sources you can go to get this information, et cetera. So I think there are ways we need to use social media. That's just one example. Social media is a much better tool for reaching folks, particularly younger folks, to educate them about health, to give them, because the other powerful thing about this genomics revolution is it's more and more about you as an individual. And it, I think, enriches people's health to be able to understand themselves, to be able to take that to their healthcare encounters, et cetera, to put more and more of the responsibility, perhaps, but certainly the ability of the individual. So I think we need to be using many more, again, creative, new media and other kinds of ways to do that, to really educate people in a very different way than the sort of health education classes that many of us had in junior high school.